Hello, Assalamu alaikum. In this video, we will understand about the mucosal immune system in our gastrointestinal immunology. It is also known as the gut immunology. So basically, let's understand about the outlines for understanding better. So the lamina propria contain large number of T cell, and basically the lamina propria is below the epithelium, and the IgA producing plasmocytes are present, and the macrophage and dendritic cell. A dendritic cell sample the lumen compartment by extending their dendrites, migrate to the mesenteric lymph node through the lymph node and present antigen to T cell for proliferation to produce B cell, and B cell will convert into the B memory cell, and plasmocyte is also known as the plasma cell. Plasma cell produce a lot of antibodies against the pathogen. This is the female which that is contain our digestive system. Whenever it will take food with microbes, it will activate the mucosal immune system. This mucosal immune system activation due to the dendritic cell will recognize the antigen. After the sampling of the antigen, we go into the innate immune system. Let's understand about bit digestive system. This esophagus, stomach, small intestine, large intestine contain ascending, transverse, descending, sigmoid, and the small intestine is the duodenum, ileum, and jejunum, while the here is the rectum and anus. Anus is used for the defecation, while the esophagus is used for the ingestion. Let's Here is the lumen inside of the digestive tract and the epithelium. This is the lamina propria, which does contain immune cell in the form of Peyer's patches. This is the Peyer's patches, and this is the epithelium. Below the epithelium is the Peyer's patches. This Peyer's patches is, and here is you can see this the mesenteric lymph node. This mesenteric lymph node and this mesenteric lymph node, while here is the mucous membrane, the glycocalyx layer, which that is the barrier immunity for protection against the invasion of the bacteria, while here is the M cell is used for the secretion of the mucus, and this is the subepithelial dome. This is the inductive compartment used for the induction of the immune response. This follicle contain many follicles contain B cell and T cell is the as a innate immune cell, adaptive immune cell. This is the microbiota. It is also known as microbiome. Microbiome contain, whenever we will ingest food, some microbiome will digest the waste to produce short chain fatty acid is known as the mutualistic bacteria, while some is commensals, neither harmful nor helpful, while the some is pathogenic. Here is the tight junction for the barrier immunity strong, for making strong immunity, and in this way the mucus is a supplementary barrier immunity. This is the isolated lymphoid follicle are also present, which that also contain a dendritic cell. So B cell and T cell are also present for the B memory cell and the T memory cell also for quick response. But anyhow, this is the dendritic cell are present in the villus, and basically where the villus are present, and this is the dendritic cell are present in the Peyer's patches, and this also the macrophage and neutrophil are present, which that will detect through antigen representing cell. The dendritic cell will recruit the T cell to recruit the B cell for production of antibodies in the follicle of the Peyer's patches to produce a lot of antibodies. But this is the inductive compartment, and in this way the B cell recruitment through production of cytokines more in this the inflammation when progress. Anyhow, this will lead to produce antibodies, and the plasmocyte basically produce for production of antibodies against the pathogen. This is the pyres patches, it is known as, because discovered through pyre. This is the dendritic cell will recognize the pathogen and activate the T cell in the isolated lymphoid follicle, and in this way also basically the B cell and T cell activation to produce a lot of antibodies. This is the plasmocytes after the proliferation of B cell to produce B memory cell and plasmocytes. Plasmocytes produce IgA antibodies against the pathogen, especially and specific. Specific antibodies produced for a specific pathogen, but it will live throughout the life whenever it will be activated by one time after the infection or something. This is the paneth cell and this is the crypt. Paneth cell is responsible for the production of mucus and antimicrobial protein and defensin. In this way, the cationic antimicrobial protein will protect against the pathogen in the lumen during the digestion and during the eating time. Anyhow, the effector compartment is this, while the inductive compartment is this. In this way, the effector compartment produces a lot of antibodies through plasmocyte or plasma cell after the proliferation of B cell. So it comes from the dendritic cell to the B memory cell and plasmocyte, and in this way it will go into the intestinal high endothelial vessel, come from the mesenteric lymph node of the lamina propria, and in this way it will produce a lot of plasmocytes, which that is the effector compartment to effect and produce the antibodies against the pathogen. Anyhow, this is the crypt and the panel cell is used for the mucus production, antimicrobial protein and defensin. Let's understand about the brush border cell epithelium. This contains brush border microvilli, and basically the villi is the bigger and microvilli is the a little fold in the cell. And in this way, in intraepithelial lymphocytes are present whenever the infection will lead to intraepithelial cell will lead to intestinal cytokines production, apoptosis, and inflammation. Inflammation will increase. And in this way, the apoptosis will occur whenever the infected lymphocyte will also produce the interleukin 15. This interleukin 15 will inhibit the T regulatory cell, will lead to cause autoimmune disorder like the celiac disease. While here is the pathogen, for example, again we will understand via summarization. And in this, the lumen, the dendritic cell will recognize the antigen as the sampling will go into the T cell in the mesenteric lymph node will through MSC class 2 molecules, and the T cell receptor R1, I don't know, but you can understand just MSC class molecule. And in this way, the major histocompatible molecule. The major histocompatible molecule through T cell after the proliferation of naive T cell to produce CD4 will lead to naive B cell activation to produce plasmocytes, and B memory cell will produce a lot of antibodies, but the B memory cell is used for the again arrival of the pathogen to quick response. And immunoglobulin already present in the mucous membrane after the one activation shot. So let's begin to understand the major problem is that our digestive system continuously exposes the pathogen and microbes due to contamination, environmental factors, allergens. Our digestive system works to evolve for survival. So that is why for surviving this continuously pathogenicity inhibition through immune cell activation, so the more eat will more inflammation, and when you will less eat, the inflammation will be decreased and the working of the immune cell will be decreased. So fasting is very important for the protection for the gut microbiome as well as the immune system inflammation down regulation. So thanks for watching. If you like my video, please make sure to subscribe, like and share. In this video, we have discussed about the mucosal immune system, the gut immunology. So thanks for watching. See you in the next video. For this time, bye. Allah Hafiz.